Hey everybody, Mr. B here, and welcome. This is our new unit, Kingdom Animalia. We're jumping to our last kingdom of the year. We've been studying a lot of small things. We've been studying bacteria, and we've been studying other single-celled creatures like protists, some multi-celled protists as well. We've studied the fungus and eaten them and watched them grow. And now we're going to be studying Kingdom Animalia, one that's probably the one that's most well known to you. And so let's jump right into it here. Let's pray first. Lord, thank you for this new week, new day. I pray blessings on our Logos at Home endeavor. I pray that students would get a lot of time outside enjoying your creation, and that things would they would be challenged, but that they would rise to it. And we thank you for all that you've done for us. In Jesus' name, amen. Okay. So let's jump right to the shared screen. I'm going to be using this system here to share a screen with you, and also to I can share my PowerPoint as well as I can share the whiteboard. And so no one wants to see a huge blow up video screen of anyone, definitely not me. And so let's jump right into it here. Kingdom Animalia, and some general features you know from your textbook. This follows along with chapters 20 through 22. You should be reading those chapters in your handouts. They will be in your locker, a printed handout of those chapters week by week. So you'll go to your locker this, hopefully you've already done that and are here sitting with those chapters now. Chapters 20 through 22 for week of March 30th. Okay, so what are we talking about? The distinguishing features of animals. How are they different than the other creatures in other kingdoms? We're going to be talking about symmetry, which is a really interesting concept, and also starting to look at various invertebrate phyla, get a sense for the diversity magnitude of these creatures, and then looking at two distinct phyla. So what do we know about animals? We know that they are eukaryotic. They are definitely eukaryotic creatures. They have complex cells with membrane-bound organelles, nuclei, and all of those other important organelles that are present. And this is important as creatures get larger and larger because cellular communication is critical. You can't have a multi-cell creature without a lot of cell communication. There's a lot of transport between cells. There's a lot of communication signals between cells. And what we get, along with increased complexity, as we go from bacteria to protists to fungi to plants and animals, we're getting increased complexity. And so we have an increased need for organization. And so one of the ways that we see this increased organization is through cell differentiation. An example of cell differentiation is development of all these specialized cells and tissues that come along with them. We haven't really seen that till now. We don't see that in the fungi. We don't see that in protists. In fact, that's one of the main ways we distinguish between algae and plants, is that algae, like a kelp, even though there might be some features that are distinguishable there, one of those nematocysts that is inflated, or the blade, or the holdfast, you can recognize those features, but if you drill down and look at the cell structure, the cells are really all the same. And we don't see that in plants and animals. We have specialized tissues, and you could just list the number of different tissue types in you or in a plant. In a plant, we have leaves and a stem and roots and fruits and flowering parts, and all these have very different cells. So we have a lot of cell differentiation. That is distinctly something that's found in animals and also in plants, but to a very high degree in animals, cell differentiation. And we'll talk about that process in going from one of these quote-unquote pluripotent cells, a cell that can differentiate, into all these other lines of cells, because in the beginning we were all zygotes, right? And so we were one-celled creatures. And so all of the information to cause that differentiation had to be present inside that pluripotent cell. And so you've heard the term stem cells. That gets at cells that are able to differentiate. Some other features, kingdom Animalia, the lacking of cell walls. That's relatively new. We do see cell walls in fungi, the chitinous cell walls, that polysaccharide that's also makes up crustacean exoskeletons and insect exoskeletons. We'll talk about them. And of course, creatures that are heterotrophic. So this fits right along with the introduction of your text on page, grab it here, right at the beginning of chapter 20. And the last one I didn't add yet is motility. So the last one, and that list is right there on 255, are that all animals have motility. And motility means they're able to, as your author says, wriggle, okay? So they're able to move to various degrees. So these are the major characteristics of kingdom Animalia. All right, let's take a look at an important concept that most phyla, we say most a lot, we qualify everything because biodiversity rules and our rules tend to fall apart quickly. We talk about symmetry a lot in kingdom Animalia. Most animals show symmetry of some kind. Most animals show symmetry of some kind. That doesn't mean that other creatures don't also show symmetry. Yeah, plants show symmetry, and there are some other organisms that show asymmetry as well. But look at this image. It's a lovely photograph of the Taj Mahal. It is remarkably symmetrical, isn't it? So think about this term symmetry. What does symmetry mean? And then let's start looking at a few creatures and thinking, well, what kind of symmetry does this organism show? So come up with a working definition. You can pause me whenever you want. Come up with a working definition of symmetry. Maybe even write it down. And then see how that turns out as we start talking about different creatures and their body plans. We're applying this concept to body plans of organisms, right? And this is a way that a complexity is organized. A symmetry is a helpful body plan tool that the Lord has used to design his creatures. And we, of course, use this all the time in engineering. And so one of your favorite microorganisms and mine, name it now, you know this one, you remember this one, is the Volvox. So there's Volvox, a couple of Volvox colonies, and we know that each one of those little dots on the outside is actually an organism with two flagella that are attached to other individual Volvox. And so there's this outer colony and there's daughter colonies inside. So where are the lines of symmetry? Where are the lines of symmetry for this organism? And I would say, and I'm remembering that I needed my draw tool right about now. See if I can easily find my draw tool. I don't know if I'm going to easily find it right now. Might have to go to the whiteboard here to illustrate. So think about the lines of symmetry for this creature. This is Volvox. Next creature. So keep that one in mind. Volvox, lines of symmetry. Next one. What are the lines of symmetry for this creature? You should recognize this one. This is in one of the first two phyla we're learning about. How would the lines of symmetry for this creature differ from the lines of symmetry from the Volvox? See anemone here. And then a third type of symmetry. Where are the lines of symmetry for this creature? And this one might surprise you. Okay, well, the lines of symmetry for the first group are called spherical symmetry. We call this spherical symmetry. And I'm going to go to the I'm going to go to the whiteboard to illustrate this. I might be able to make it work here. Oh, it looks like I can get it to work. Good. So there's a line of symmetry. We'll do another one. Let's make another line of symmetry for this orange. Let's go this way. You can hold the orange in any fashion and make a line of symmetry. There's no top, there's no bottom, there's no left, there's no right. We can keep on going here and make pretty decent lines. These aren't perfect, but you get the point. These are pretty close. This is called spherical symmetry. And that's the kind of symmetry the Volvox has. That's the kind of symmetry the Volvox has. Now, sometimes you're going to have a type of symmetry and you're going to say, well, this isn't precisely, there's 4 daughter colonies on this side, there's 4 daughter colonies on this side. But if you took any daughter colony and divided it, it would also have spherical symmetry. So spherical symmetry, any plane through the center of the organism results in 2 identical halves. So now you have a new question to ask the creature that your neighbor brings to your doorstep. All right. What kind of symmetry do you have, sir? It's a Volvox, and he says spherical symmetry. Indeed. 2 identical halves, any plane at all. No top, no bottom, no left, no right. Okay. Next type of symmetry, our sea anemone, radial symmetry. Radial symmetry means that any longitudinal plane through the center, any longitudinal plane through the center. Longitudinal means along the length, like this. So we'll move that a little bit to the left for you who are really design people and you're noticing my lines aren't perfectly straight, or they're not perfectly square, I should say. All right, that is a line of symmetry for a radially symmetrical object or creature. All right, and so looking down on the sea anemone, think about sitting on a dock, looking down, there's a jellyfish, and you just see the top of it. You can slice it any way if you're looking down, okay? You can't slice it any way you want if you have a profile view. So these creatures have a top and a bottom. These creatures have a top and a bottom. And you're gonna see we have certain phyla that are radially symmetrical. In fact, a lot of them are radially symmetrical. So this is considered an increase in complexity. If radial symmetry is needed, then this creature is going to be a little bit more organized. It's going to need more organization than, say, a spherically symmetrical creature. Okay. Third one, our earthworm. Just like this cute friend looking at us here, this little, I thought I had the name of that dog for, we used to have one that looked a lot like that. Border Collie, looks like a Border Collie. Plane of, or line of symmetry for this Border Collie. We'd never want to cut this Border Collie along its median here, as they call it. But that would do it. And that's pretty close, right? Again, no creature is absolutely perfectly atom for atom, cell for cell symmetrical, right? You can see that the nostrils are not perfectly symmetrical. You can look in the mirror, and that might be something fun to do, is look in the mirror and realize that you are considered symmetrical. You're actually considered bilaterally symmetrical. Only one line will cut you into 2 halves, but you know that that's not perfect even. Superficially, it's going to be symmetrical, but you know based on the internal organs you have, your liver is not going to be divided exactly right. Your heart is going to be on one side. Your appendix is going to be on another side. You know about the big wart on your big toe that no one else knows about, right? And so these are general body plan type of statements. These aren't precise atom for atom, cell for cell statements. So these are the 3 major types of symmetry we'll be dealing with spherical, radial, and bilateral. Now, most of the creatures we're going to be looking at are radial and bilateral. Okay. I'm not going to try to draw a cut for that earthworm. And you might be surprised that that earthworm's bilaterally symmetrical. They actually are bilaterally symmetrical. They have incredibly complex body systems. They have many, many veins and arteries. They have a brain. They have reproductive organs. And so they can only have one cut as well. And we were going to dissect an earthworm. We're not going to be doing that here now, but we will have some fun virtual dissections for you guys. And part of that is learning the anatomy of these creatures by their symmetry, and also with some other terms we're going to look at here in just a moment. So here's some more examples. Here we see examples of radial and bilateral symmetry. There's our symmetry. There's our sea anemone again, your pot for a plant. Your crayfish is bilaterally symmetrical, and your shovel is as well. And because you're going to ask the questions, I tried to think what questions will they ask, and one of the I thought you might ask is what other types of symmetry are there? Here we go. Supersymmetry, I'm calling this. And that includes creatures, quote unquote, like viruses from our Virus Explorer activity. And we see that we have icosahedral. If you remember exploring and looking at icosahedral, that means a polyhedron with 20 faces. So an amazingly symmetrical, complicated symmetrical creature, the adenoviruses. And then also our friend the Ebola virus, which has a helical symmetry. So helix, RNA, DNA are helical molecules. They have a helical symmetry. And so that's another very complex type of symmetry that we're not going to see in Kingdom Animalia. Okay, the last few minutes I want to spend time just reviewing and learning some new anatomical vocabulary. So when you do your virtual dissections, when you describe a creature, you need to use the anatomical terms that relate specifically to that part of their body. And so because we have so much organized complexity now we have the need for more terminology. We don't just have membrane versus non-membrane bound organelles. No, we have distinct regions made up of differentiated cells and tissues that are different from another part of the creature's body. So bilaterally symmetrical creatures have a lot of anatomy and a lot of anatomical geography terms, really is kind of how I think of it. And so the head region is called the cephalic region. The head region is called the cephalic region. Okay, so if you're referring to head at all, cephalic is the term, and that's also considered cranial. And you're moving in one direction or the other. You're moving from cranial here to more and more cranial as you move from right to left. And you note here in the middle, you can see that where the cranial and caudal division are. Right? So a lot of times we're not really right on the head, but we're closer to the head than we are to the tail. And so these terms are really helpful for the in-between places as well. They're not just used to describe that specific region. So the cranial or cephalic region, and then the tail region is the caudal region, caudata. That's our salamanders, creatures with big tails. So caudal fin in a fish is the tail fin. Okay, and then anterior, you know that one already, toward the front. So the anterior is going to be towards the head region, the chest region, the front legs of this creature. Posterior, the reverse of anterior, it's going to be the tail and the back leg portion of this creature. There's some extra terms on this diagram you're not going to need to know, but they're all useful, and you've probably seen these if you've taken any physiology and anatomy type classes, if you're taking that elective this year. Dorsal and ventral are very important when we do dissections. Dorsal is the back, and so the back of this dog. A dorsal of a stingray is going to be that flat top part. The dorsal fin of a shark is the back region. Your dorsal, which is kind of interesting because we're upright creatures. We're up on 2 legs, not on 4. Our dorsal is really the same as our posterior side, and our anterior is our ventral. So ventral is the belly. So the belly of this dog, the belly of a crayfish, and earthworms have these same distinctions because they're bilateral as well. Okay, a few more terms here. Lateral means the sides. So when we say the lateral of the fish, we're looking at the sides of that fish where we see the gill cover and gill openings. We also see one eye, and you can get a nice kind of profile view. The profile view, that's lateral. And medial, the medial view or median would be right down the middle. So the median in a bilateral creature is right down the middle. And there's that median, divides right and left. And then the last one that I need you guys to know is transverse. And transverse means perpendicular to the midline. And so in a tree, for example, a tree with tree rings, I'll bring mine over here and show you real quick. That's a transverse. This is a transverse cut. So if you're thinking dissection or if you're thinking surgery, that's a transverse cut there. You can see the tree rings really well. Okay, and the transverse cuts here on this this creature are the ones, there are 2 transverse cuts, right, that are shown. One is for the whole body. And so this is a transverse plane, right? It's perpendicular to the midline. There's the midline cut, and then right down the torso would be transverse. And also we can get a transverse cut over here of the leg. The midline of the leg is going to be running up and down. That's vertical. Transverse is going to then be horizontal. So transverse cut runs across, perpendicular to the midline. So we're going to be using a lot of these terms, especially when we talk about larger animals. But at least ventral and dorsal, anterior, posterior, caudal, and cephalic or cranial are very important in many invertebrates as well. And so to get to the big biodiversity tour intro here, we are going to be looking at a lot of invertebrates this week and for the next couple of weeks. Invertebrates make up 95 to 97% of all animals. So something like 1.25 million identified species are invertebrates. And just really coarse definition would be no backbone. We'll get into the specifics of that once we talk about vertebrates. There's a little bit more to vertebrates than just a backbone, but they do have a backbone, and they make up about 3 to 5% of all animals. And so 36 total phyla. We're just sampling a few. And here are those phyla. The major phyla, and I'm saying major by number. So I've listed 1, 2, 3, 4, 5, 6, 7, 8, 9. You don't have to memorize these phyla. We'll be going through phylum by phylum, a select few of these, 5 of these phyla. I listed these because they're so numerous. These tend to have numbers of species in the thousands. And so they're considered really important as far as diversity and just importance ecologically and to people. So the phylum Porifera, the sponges, phylum Cnidaria, that's a silent C. The jellyfish, coral, and anemones, those are going to be our first 2 that we're studying this week. Phylum Annelida, 3 big phyla of worms. And we're not going to get into the details of the worms. We're going to be putting them aside. We'll learn some peripherally about them, but just to adapt to our schedule and some lost time, we're going to be moving past the worms fairly quickly. Phylum Annelida are the segmented worms. You notice the segments on that earthworm Also include polychaetes, which are a really cool marine tube worm, and then leeches, everyone's favorites. You can find them in Paradise Creek. Okay, and a great time to just be doing some of your own biological sampling here. Nematodes are the roundworms. Most of them are considered parasitic and pathogenic. You have to give your pets shots so they don't get roundworms of any type, heartworms, etc. And then the flatworms, which include some really amazing creatures like planaria, and also some other creatures like tapeworms, which are also parasitic. Phylum Mollusca, we will be studying Phylum Mollusca, the bivalves, clams and mussels, snails, and the octopodes are the major creatures there. Phylum Echinodermata, really neat type of symmetry we haven't touched on yet when we look at them. Starfish, sand dollars, and sea cucumbers. Phylum Arthropoda, this is the largest group of animals, period, the arthropods, spiders, insects, crustaceans, and many more. And then the moss animals, which you may not have heard of, the bryozoans, really microscopic animals, and numerous species there as well. So that's the big picture overview. Here's a little bit about their numbers in case you're curious. And so all of these species, several thousand at least, but notice the big ones here. Phylum Arthropoda, look at that, 1.1 million. Around the same for the nematodes. Lots of little parasitic roundworms out there. And some of these other ones might surprise you. 5,000 species of sponges, 11,000 species of cnidarians, over 100,000 species of mollusks. And so the ones that we're going to be looking at are these 5. The sponges, the cnidarians, the mollusks, the echinoderms, and the arthropods. So I'm looking forward to exploring these creatures with you guys. Expect a lecture on the poriferans and the cnidarians this week. This is an introduction to Kingdom Animalia and an introduction to the invertebrate phyla. And so we'll be getting into this broader group, the invertebrates, here really soon, which include this fabulous beast, and which includes our first phylum coming up, the sponges. So I look forward to introducing them to you, and guys, we'll see you next time.